So without further ado, let me introduce our amazing speaker. Kathleen Cusick is an assistant professor in the Department of Biological Sciences at the University of Maryland in Baltimore County. She does maintain her Florida residency, of course, and her home address is actually here in Indian Harbor Beach, where she lives with her husband, a stone's throw from the Indian River Lagoon. She received her master's in marine biology from Florida Tech, Florida Institute of Technology, and did research on bioluminescent dinoflagellates at Harbor Branch Oceanographic Institute. She received her PhD in microbiology from the University of Tennessee, where her research focused on the effects of the toxin that these types of dinoflagellates produce. It's called saxitoxin. Some of you may have heard of it because it's what we want to avoid with fish when we eat fish. So her lab uses really cool molecular tools to study the microbial ecology, because these critters are teeny tiny, and she's working in different coastal marine systems. But she's focusing the talk today and her study on the dinoflagellate Pyrodinium bahamense and the bloom ecology. So this particular critter is what makes the waters glow right now when everybody wants to go out and see the amazing algal blooms at nighttime and see how beautiful the bioluminescence is. So it's a good and it's a bad, and she's going to talk about that. She's married. For fun, she likes to run and swim in open waters. She loves the Shenandoah National Park, but she wanted to let you know that she loves swimming at the Gleason Park pool as well. And with that, I'm going to turn it over to Kathleen. Thank you so much, Kathleen. Let's give her a round of applause. Thank you, Lisa. Really nice to hear.
All right. Thank you, everyone, for being here today, and those of you on Zoom. So, I'm sorry, Lisa, I think you took my phone.
Oh, I did.
She's trying to walk away with my iPhone. And I only say that because I have created this. Well, I was going to use it for timing. Okay. So don't worry about it. All right. That's okay. We'll just get started. So I've made this talk very free-flowing because I want you guys to be able to ask questions. I know you're going to be asking questions in the chat as well then. So let me just get some semblance of time here, though. All right. Here we go. So I'm going to be talking to you today about Pyrodinium bahamense, which I consider the Reese's peanut butter cup of harmful algal bloom species. And disclaimer, as you could tell from my short bio, I am a molecular microbial ecologist. So this talk is going to incorporate some DNA and genetics as well. So again, give me feedback. Let me know if something I'm saying is not clear to you all. So first of all, Pyrodinium bahamense, Reese's peanut butter cup of harmful algal bloom species. I want to start out by giving more of a broad overview on harmful algal blooms, because there's a lot of them here in Florida, right? And there's a lot up in Lake Erie. They're in marine and freshwater systems. And so with these HABs, you can have different species and different toxins. So, and again, they're produced by dinoflagellates, which are microbial eukaryotes, and cyanobacteria, which are often referred to as blue-green algae. And these are just some representative images of different harmful algal blooms. We're familiar with Pyrodinium bahamense, which is in the Indian River Lagoon. And then this is just also showing you, though, that Pyrodinium produces saxitoxin. Well, so does the blue-green algae Lingbia, which is a problem in freshwater systems. This is a lake in South Carolina that gets these huge mats of Lingbia. And then you're also probably familiar with the microcystin bloom in the St. Lucie estuary several years ago, right? Which was thick green soup. So you have different species and they produce different toxins. So I want to give you a little bit more background on dinoflagellates, since Pyrodinium is a dinoflagellate. They come in all shapes and sizes. You could see them under a regular compound microscope. They range in size from 5 microns up to 1 millimeter. And you can see they have this really fascinating differences in shape. So these are just different species of dinoflagellates that I'm showing you up here. Pyrodinium is over here, highlighted in blue. But what I want to make clear is that they're single-celled microbial eukaryotes. And so when I say the word eukaryote, who is familiar with what a eukaryote versus a prokaryote is? Okay, eukaryotes, you are all eukaryotes as well. Our cells have organs and compartments in them, as opposed to bacteria, which don't have all of those compartments and organelles. So dinoflagellates, eukaryotes like you and I, they're part of the phytoplankton. Some of you may be familiar with the terms phytoplankton, zooplankton. Zooplankton would be the copepods and the other little critters that are around out there. And one other thing that I want to impress upon you is that these guys, which are, Pyrodinium is 40 microns in size, right? That's less than a millimeter. They have huge and complex genomes. Dinoflagellates have more DNA in their cell than a human cell, and it's a lot more complex. It's a mess. Whereas human DNA and other mammals and the DNA of other organisms in general is neatly organized. It's wrapped around these proteins. It has genes that are arranged in pretty good order. Dinoflagellates, huge amounts of DNA, and it's unorganized, and they can have hundreds of copies of the same gene, all with slight variations. So nowadays, there's all these different companies that will sequence your DNA, right? And so we can do that for the human genome. But with dinoflagellates, if you look in some of the databases that are out there, there are only a few that we have genome sequences for because they're so massive and complex. All right, a little bit more about Pyrodinium bahamense. It's often referred to as a thecate dinoflagellate. If you look at it, it has this armoring around it. You also have naked species of dinoflagellates, or athecate. They don't have that tough cellulose wall around them. As I said, it's about 40 microns in size. Pyrodinium bahamense, you may be familiar with it because we have it here in the Indian River Lagoon and over in Tampa Bay, but it is also found in the Indo-Pacific, where there are red tides, toxic red tides of it very frequently. We know now that in Florida, it is toxic, and I'm going to discuss that in a few slides. And then it also occurs in Puerto Rico because one of its traits is that it's bioluminescent. And so you are familiar with the bioluminescence at night out in the Indian River Lagoon. The same thing in Puerto Rico. How many of you have ever been to one of the bioluminescent bays in Puerto Rico? Okay, they're pretty spectacular, right? You take them out kayaking at night and see all the bioluminescence. That bioluminescence is from Pyrodinium bahamense. The toxin that Pyrodinium produces is saxitoxin. And then the saxitoxin has what's called derivatives or congeners, different forms based on some other atoms that come off of it. Collectively, they're referred to as paralytic shellfish toxins. And I will talk a little bit more about that in the coming slide as well. The closest relative to pyrodinium is alexandrium. And I bring that up just because those of you that study harmful algal blooms may be very, very familiar with the alexandrium genus. Woods Hole, Don Anderson up there, they do a lot of studies because alexandrium is a big problem up in New England. They have to monitor their shellfish industry as well because it also produces saxitoxin. So again, I bring up the name alexandrium because some of you, if you have ever been up in New England, in Massachusetts, might have heard this name. A little bit more about the two traits that I'm going to be talking about with pyrodinium, toxicity and bioluminescence. So dinoflagellates can do a bunch of different things, right? But toxicity and bioluminescence are two of the best known, right? You hear about these toxic, harmful algal blooms, or you can see the bioluminescence, which there are other bioluminescent species of dinoflagellates as well. So overall, when we talk about harmful algal bloom toxins, there's 17 major types. And then when you look at dinoflagellates as a whole, all the different species, in the marine environment, there's about 1,500 marine species of dinoflagellates. And of these, about 70 of them are bioluminescent and about 100 of them are toxic. So really, that's a small percentage of the overall dinoflagellates. And of the toxic ones, only about 12 are bioluminescent. So it's a kind of a small niche, but in general, it is some of these toxic bioluminescent species that you hear the most about. Off the coast of California, they have Pyrodinium blooms, toxic and bioluminescent. Alexandrium, toxic and bioluminescent. Pyrodinium, toxic and bioluminescent. What I'm showing you here is a phylogenetic tree. You don't need to see all of the species names. It's just showing you that these traits are distributed among all of the different dinoflagellates in this dinoflagellate tree of life. All right, Pyrodinium bioluminescence. I'm speaking to a crowd that has gotten to see Pyrodinium bioluminescence firsthand. And I won't go into the whole reaction of bioluminescence, biological reaction that occurs within the cell. It needs a compound to work on. It needs a specific enzyme known as luciferase in order to generate this blue light. Now, another example of bioluminescence, can anybody give me a terrestrial example of bioluminescence? Right, exactly. Fireflies. They have a different system, but they're producing bioluminescence as well. So it's the same overall reaction. Well, this was supposed to be a little video. Here we go. This is just some bioluminescence up at the Hollow River Canal at night from the kayak with a net. All right. Now I want to talk a little bit about the other trait of pyrodinium, which is saxitoxin. Don't fear it. It's a fascinating compound. As with these different HAB species that produce different toxins, in these different toxins, you have some that function as neurotoxins, which saxitoxin is. Another example would be brevetoxin. Those of you that are familiar with the cariniatides over on the West Coast, they are producing brevetoxin, also a neurotoxin. And so these, you can see that some of these are pretty big molecules that these little guys are producing. Saxitoxin is fairly simple. How these neurotoxins work is that they are targeting voltage-gated ion channels in other mammals, right? So they're going to target the sodium channel, the calcium channel, the potassium channel. And as I had mentioned earlier, saxitoxin is the parent compound in this group of molecules that are collectively called the paralytic shellfish toxins, PSTs. And this is just, if you had to describe its structure chemically, it's a perhydropurine alkaloid, so fairly simple. And then what this is showing you is a table with this basic structure. We're not going to do chemistry here, but you have different groups that can be coming off the parent structure that make the different derivatives. Now, saxitoxin is the most potent of this entire class of paralytic shellfish toxins. But scientists use these different congeners to study different strains, because you will find that strains of pyrodinium in the Indo-Pacific are producing a certain collection of these different compounds, whereas in the Indian River Lagoon, it's going to be a different collection. And then Alexandrium is going to be producing yet a different collection. And I simplify that because you have differences even within strains of the same species. So paralytic shellfish toxins. how saxitoxin hurts you and I. It causes paralytic shellfish poisoning and pufferfish poisoning. So what's happening is that you have these cells out in the environment, and then you have the filter feeders, the clams, the oysters that are taking in the cells that are producing the toxin, and they're bioaccumulating that toxin in their tissues. And then you eat those clams or those oysters, and that's when you get the big dose of the toxin. And big dose is not really a big dose. 200 micrograms, right, which is less than a milligram, is lethal to the average weight human being. But you're going to need to eat a lot of oysters that have accumulated a lot of the toxin in their tissues. And then pufferfish poisoning is very similar, where you have the pufferfish accumulating the toxin within their tissues. So it's bioaccumulating so that when you eat this organism, you get the big dose of it. So in general, when you're out there in the water and you're swimming with pyrodinium, that toxin, most of it is retained within the cell. So it's not like you're swimming in this pool of saxitoxin. And this is also a map just showing you how paralytic shellfish poisoning events have spread worldwide. Now, this can also be attributed to, we now have better monitoring programs, right? But in general, harmful algal blooms are increasing, and we can get into climate change. We can get into nutrient input, right? Which we're all familiar with here in Florida. But in general, these paralytic shellfish poisoning events and HABs in general are increasing worldwide on a global scale. Saxitoxin, I just want you to understand how it's affecting you, because when you eat it, you would be getting some tingling in your extremities, tingling around your lips. Yes, you could have some diarrhea. That's a similar symptom. A lot of the saxitoxin symptoms are similar to brevetoxin or some of the other neurotoxins, right, in terms of the diarrhea and the nausea. But the tingling, and then eventually respiratory paralysis, you might have trouble breathing. And that's because saxitoxin is binding to this sodium channel. And I won't get into the domain architecture of the sodium channel. Suffice to say, saxitoxin has, when it's there in the cell, it has a positive charge on it. And the sodium channel, which it's a protein, proteins are made up of amino acids. Some of the amino acids that are here at the core of the channel have negative charges. So saxitoxin can bind with them very well and block the passage of sodium ions going into the sodium channel. And then that's why you're experiencing this tingling. A little bit now about saxitoxin biosynthesis in dinoflagellates. Remember, I told you, dinoflagellates, massive complex genomes. We are lucky with saxitoxin in that the core toxin genes have been identified. And I won't go into a lot of detail on this, but a group in Australia did wonderful work to identify these genes in the different dinoflagellate species that produce the toxin. And there is the SXTA gene that starts the reaction, that's going to start making the saxitoxin molecule. This protein, and these bubbles here are indicative of this SXTA protein, right? And I just want to make clear to everyone out there that proteins, we need all of them. That's what's making your hair color brown. That's what's making your eyes blue. It's the proteins that are being produced in your body. So SXTA is the enzyme that starts, the protein that starts this reaction, building the molecule. And proteins can be very large and complex. And they have found that there's two different forms of this saxitoxin SXTA protein. One that has 3 domains, which are part of the protein, SXTA 1, 2, and 3. And then there's a form of the protein that also has SXTA4. So remember the term SXTA4, because this portion of the protein, which is coded for by a gene, right? All proteins, you need to have a gene sequence for in order to make it. So SXTA4 is a gene, and it's specific for toxin biosynthesis. So we are using it as a molecular marker for the genetic capacity for toxin biosynthesis in Pyrodinium bahamense. If it doesn't have SXTA4, and this is, for the different species and strains that are out there, then it's not producing the toxin. If it has the gene, it has the potential to make the toxin. Now, whether it actually makes it or not, this also comes into play. There's no easy answer for that. But SXTA4, remember the name of that gene. Saxitoxin outbreaks in Florida. Some of you are probably very familiar with this when it occurred. So pyrodinium existed for years in Florida, right? With no known record of toxicity. And then in the mid-2000s, people across the United States got sick eating pufferfish harvested from the Indian River Lagoon. And saxitoxin was determined to be the source, coming from Pyrodinium bahamense. So up until that time, you had these great blooms of pyrodinium, you had the bioluminescence, but you didn't have the toxic outbreaks that we have now. And so how did Pyrodinium bahamense become toxic, right? It's been there, been bioluminescent, not producing toxin, and now bam, now it's producing toxin a lot, right? And there's a permanent ban on pufferfish harvesting. And this was back in the mid-2000s, and I was doing some field work with pyrodinium there. And here is an aerial shot of Patrick Air Force Base. And I talked to some of the pufferfish fishermen, and I don't know, maybe some of you are out there in the crowd now, and they know their history of saxitoxin. There was construction going on at Patrick Air Force Base, and saxitoxin is very potent, and it was used by the government in the 70s to make suicide capsules to give to their pilots if they got shot down over enemy territory. And so the fishermen were convinced that the government had not given up all of their stockpiles of saxitoxin, and that some had escaped from Patrick Air Force Base out into the Indian River Lagoon. So in any case, however that initial toxin got out there, we now have pyrodinium toxic. And here again is the title of my talk. It's the Reese's peanut butter cup. I consider it, because what are Reese's peanut butter cups but two great traits, chocolate and peanut butter? What is pyrodinium but toxic and bioluminescent? And so I do have some Reese's peanut butter cups for you all. And then questions during the talk, if I see you falling asleep, I will be chucking some out there as well. So, and some of them are pretty big Reese's peanut butter cups. So it's toxic and it's bioluminescent. It's chocolate and it's peanut butter. And so, no, and in fact, I should have gotten you dark chocolate Reese's peanut butter cups, because dark chocolate, better than milk, it contributes to a better gut microbiome. So, but in any case, so why are some blooms toxic and others not? And this can apply to Florida, but also pyrodinium and alexandrium and other HAB outbreaks. For example, off the coast of Mexico, where there's toxic pyrodinium blooms, they isolated 9 strains. And what that means is they went out in the water, and from the same water sample, they picked 9 single pyrodinium cells, and they got them to grow up in the lab in culture. So each of those single cells divided and multiplied, and that's how we got the strain. And of those 9, 8 of them were non-toxic. Only 1 was toxic. And they found this with other strains and species and HABs as well. And a lot of the studies look at whole water samples where they're taking all the cells and extracting the DNA and the RNA and looking at toxicity, or they're based on lab studies. There's a lot of lab studies out there that like to work with the toxic species, and they change the different nutrient levels. They add more nitrogen. They add a different nitrogen source. They change the salinity level because that affects the amount of toxin that's produced, the congeners that are produced. But what we did, because molecular tools for dinoflagellates have been lacking in comparison to bacteria and human because of their large genomes. But now we're getting it. We have next generation sequencing that we can use for things. We're making a lot of probes. So molecular analyses are becoming more common. And we need these because if you look under a scope, all the cells look very similar. And you can't tell if one is toxic or not just by looking at it under the microscope. So there have been studies now done that say, oh, yes, within this single water sample of Pyrodinium or Alexandrium, all the same species, these cells actually have a lot of genetic diversity. But they were using these microsatellite markers, so they're other common markers. So what we're doing is doing single cell analysis from natural populations, and I'm going to talk about different sites here in the Indian River Lagoon. And then we also have a site in Vieques, Puerto Rico, it's called Mosquito Lagoon, or Mosquito Bay. Sorry, we have Mosquito Lagoon here. We have Mosquito Bay in Puerto Rico. So we've been doing single cell work. All right, I'm going to show you some molecular analyses now. And I want you to be able to understand this assay so you can then understand what I'm talking about in the future slides when I say how we are studying these subpopulations in the Indian River Lagoon and who's toxic and who's not. So we developed a multiplex PCR assay. How many of you know what PCR, the polymerase chain reaction, is or have heard the term? Okay, I'm going to go over it briefly. You have DNA. And if you have some what are called primers that are nucleotides that are similar to the DNA of interest, you can amplify that gene. So you can we go into the pyrodinium cells and we're trying to amplify their SXTA4 gene because we want to see if it's there. So it's this reaction that occurs over like 40 cycles. You have to heat it up to get the DNA to separate, the primers anneal, and you get this whole reaction where you're getting more and more copies of the gene because you want to see if it's there because you can't just, again, as I said, tell by looking at the cell, it has that gene. Typically, you're doing PCR on a single gene. Well, we are picking single cells and we're putting them in little tubes. And so if we would just do the SXTA4 assay, and we don't see a band. And this is a representative image of what the PCR reaction, like the end product of it. This is called gel electrophoresis. So this is gel, if you can imagine something like gelatin, and you have your samples that are in wells and they're being pulled through the agarose and they're being stained. So these bands are indicative of a particular gene. And so we have these single cells in the tube, and we want to see if they have SXTA4 or not. Well, if we don't get a band on the gel, because ignore these bands at the bottom, these are called primer dimers. They're just an artifact of the PCR reaction. But if you don't get anything, well, does that cell not have SXTA4? Or did I not get the cell in the tube? Or since it's coming from an environmental sample, even though we're washing the cells, are there contaminants in there that inhibited the reaction from taking place? Did I screw up adding my reagents, right? We don't know for sure. So we are actually doing 2 PCR reactions at the same time in that tube with a single cell. One that targets a very common gene, the 18S rRNA gene. You also have an 18S rRNA gene. So for those of you that aren't real familiar with multiplex PCR, right now say, wow, that's so cool that you got this assay to work on single cells from the environment in dinoflagellates, which have very complex DNA. So what we found was that within a water sample from the same location, there were some cells that had the toxin gene, SXTA4, and others that did not. So first it was developing this tool to screen for it. And again, I go back to saying we're fortunate in that we've identified the core genes in saxitoxin biosynthesis, as opposed to brevetoxin or ciguatoxin, because those, well, we just know there's a bunch of genes that are called polyketide synthases that are probably involved, but we can't say it is gene A that is helping produce this toxin. So we have been able to use SXTA4 to see whether this cell has the genetic capacity. Does it have that gene? Can it make the toxin? So then we looked at genotype frequencies among pyrodinium populations. I don't want anybody to be afraid of the term genotype frequency. I know that kind of gets into genetics, and genetics can be scary. I'm not a geneticist. I am a molecular microbial ecologist. What I'm referring to when I say genotype frequencies are when we have collected 20 cells from Lee Wenner Park, and we have screened them, what percentage of those cells have the SXTA4 gene? If, you know what, I should have done 10 cells. Let's keep it simple. If I do 10 cells and 8 of those cells have SXTA4, well, we're saying that the SXTA4 positive frequency is 80%, whereas SXTA4 minus negative, those cells don't have the gene. So does that make sense to everyone? We're going out there, we're looking at the cells that are in the water, and we're seeing who is able to produce the toxin. And for this initial, what I'm showing you here, these initial samplings was when I was first getting the assay up and running. So the sites in the Indian River Lagoon that we looked at, there was one sampling in Diamond Bay in 2019. TBM is Palomar Bay Marina. I don't know how many of you are familiar with that. It's a little blurry on the map here, but it's right down near Dragon Point. And then Lee Wenner and the 520 bridge, right? Real scientific terms here. That bridge going over 520, the causeway. Actually, it was the little bridge, is where we were sampling. So, and it was collecting a bunch of cells and screening the cells at that time. Now, I also include data from Mosquito Bay, Puerto Rico, because in Puerto Rico, the bioluminescent bays are a huge tourist attraction. And nobody has looked at whether the cells in Puerto Rico are toxic or not. And so, or measured for toxicity. And really, they don't want to know. So we looked at the single cells and we did find it. Interestingly, though, it's in a smaller percentage of the population, right? Like here at Lee Wenner, 100% of the cells, everybody had SXTA4, everybody had the toxin gene. But at some samplings in Puerto Rico, it was down to 43%. Only half the cells had SXTA4. I'm just going to briefly touch upon this. This is just showing you sequence data. Remember I had said DNA, nucleotides. Well, when you sequence your DNA, this is what it looks like. You have C's and T's and A's and G's. So we looked at the saxitoxin A4 sequences that we got from some of these single cells. And yes, we saw a bunch of variation in certain regions, which is typical of dinoflagellate genes. And then we also always consistently recovered two different sequences, some that were lacking a 15 base pair region. I won't get into all of that right now. I'm just putting it out there to let you all know we have been looking at the SXTA4 gene diversity in natural populations. All right, so now we have this tool. So we want to use it to try and map the blooms in the Northern IRL. But I'm not calling them blooms. I'm calling them subpopulations because depending on who you talk to, there's one long bloom all summer in the Indian River Lagoon, right? Because pyrodinium is there. It starts in March or April, and it's going to be at different locations throughout the summer. So I'm calling them subpopulations because we're sampling 4 distinct sites. and they're pretty separate from each other. And we want to see what those different groups of cells are doing there. So the sites that we've been looking at over the past year, we started off with Haulover Canal and Diamond Bay. Those were our two main ones. And so I'm thinking that most people in this talk are familiar with Haulover Canal. That is where, and Beacon 42, where they take a lot of the nighttime kayaking tours. So Haulover Canal is actually in the Indian River, if you launch a kayak onto that side. And then Beacon 42 is actually just on the other side in Merritt Island National Wildlife Refuge in the Mosquito Lagoon. We've just, within the past few months, added that site because it's intriguing. The Mosquito Lagoon is considered the more pristine of the different sites in the Northern Indian River Lagoon, right? You have the Mosquito Lagoon, the Indian River, and the Banana River. And so Mosquito Lagoon is thought to be more pristine. So we're interested to see how toxin levels and toxin genotypes differ, especially when these two are fairly close, right? There's the Haulover Canal that goes through and connects them. And then we're also sampling Diamond Bay and Kelly Park down in the Banana River. Just to give you a sense of what we're doing out there, we start off taking whole water samples. Now, especially for the RNA analysis, we need a lot of cells because we need to break them open and extract their RNA. So we typically filter 9 to 10 liters through this mesh that's going to be better at catching pyrodinium, right? It's 20 micron mesh. Pyrodinium is 40 microns, so it's going to get caught on that mesh. So we collect surface water, we concentrate it. Say if we collect 10 liters, we might concentrate it down to 100 mils so we can better work with it. And then with those water samples, we are doing different types of analyses. Genotype analysis. I just talked to you about that. We're picking the single cells and looking for the presence of SXTA4. We're also looking at SXTA4 expression. The cell can have the gene, but it might not be expressing it. The gene has to be turned on for the protein responsible for that gene to then be made, which is necessary to make the toxin. So if we don't see induction, if the gene is not turned on, well, then it's a good sign that even though it has the gene, it's probably not producing the toxin. So, and we're using different molecular methods to assess the transcription of the gene. I just mentioned them here for those of you in the audience that do want to discuss molecular methods with me. Then we're using quantitative reverse transcriptase PCR and digital PCR. All right. We're also looking at the toxin that's produced by the cells. We send them to a lab up in New York that analyzes that are saxitoxin experts. They do all the measurements out on Lake Erie as well, so that we can know whether the cells are producing the toxin or not. And then also, we're just doing normal cell counts. How many cells are out there? And so we're collecting a sample and counting them under the microscope because we want to be able to relate how many cells are out there to what proportion of those cells have the toxin gene, to who's expressing the gene, to what's the overall amount of toxin in each of those cells. This is just an image of nighttime sampling out in the kayak at Haulover Canal. And I just want to give a shout out to Sunny Applegate, who was a guide with A Day Away Kayaks. Last summer, they were very helpful in getting me out there at night because it's difficult to sample and be in the kayak and not end up wherever the current is taking you. So they were a big help. And that's just a red light, right? Because you don't want to disturb the bioluminescence. So let me tell you a little bit about some results. Cell abundances. Okay, when you look at environmental data, it can be clear as mud. So on these next few slides, it was a lot of work, but I'm going to try and condense it down to little bullets for you all. So cell abundance at these 4 sites fluctuates. Diamond Bay is like the hotbed for pyrodinium. It is always there. And I'm so disappointed. Sunday or Monday, not there this week. Guess it's gone for the season. So, but as opposed to Haulover Canal, which there were some sites in March where you don't find it. Okay. And then down here we have Kelly Park and Beacon 42 as well. This y-axis is showing you the cells per liter. And then along the bottom are just the different dates. So cell abundances fluctuate. So let's talk about genotype frequencies. At Diamond Bay, again, now this top graph is the cell abundances, cells per liter in the water. And then the dates align with, on those particular dates in the bottom graph, when we did the single cell analysis and looked at the genotype frequencies. And so the purple is SXTA4 plus. So if you look at this first bar, it's showing you that all of the samples in Diamond Bay from September 15th, 2021 had SXTA4 in them. And it also is just in relation to cell abundance. And then when you look and you see some with the blue, that is SXTA4 minus. So you'll see in June in Diamond Bay, well, it was about 80% of the cells that had SXTA4 and 20% that didn't. So you can see that overall SXTA4 plus is the dominant genotype. Most cells have it. But what makes a significant frequency difference? Does anybody have the answer for that? Give them a Reese's peanut butter cup, whoever does. Okay, so Diamond Bay toxicity. What we did here, again, this is showing you cells per liter, and this bottom graph is showing you the toxin quota on a per cell basis. Now we had to take the average, right? Because we cannot measure toxicity from single cells right now. This group in China did. But in any case, when I consulted with my chemist experts, right now, it's too messy and noisy. So we had to base it on what were the cell counts that day. Okay, there were 100 cells in the water, and then we factor that in order to determine what the picogram per cell was. So that's the toxin content in each cell. And so this is just showing you, again, that toxicity varies. And you have to look at this data quickly, right? So your eyes might not be drawn to the trend, but in general, toxin content increases when the cell abundance decreases. Or you could say when cell abundance increases, the amount of toxin that's being produced per cell actually goes down. So even though you're getting these blooms out there, right now, this trend with Diamond Bay was showing toxicity decreasing. Haulover Canal, genotype frequencies. Again, you're looking at it, okay, clear as mud. But really what you can see is when you look at cell abundance on the top graph, genotype frequencies on the bottom, is that SXTA4, that genotype dominates when you have less cells. And that SXTA4 minus cells begin to emerge as the cell abundance increases, as you're getting more cells in the water. Haulover Canal toxicity, it fluctuates. But if you look at this June date, June 26th, cells, high number of cells, low toxin content. Now it's difficult to see a trend right with just 4 bars. I'm going to show you one that enhances this in some coming slides. Kelly Park, again, we just started studying Kelly Park and Beacon 42 this spring and summer, so not as much data, but Kelly Park with its genotype frequencies, similar to Haulover Canal, SXTA4 minus cells begin to emerge as the cell abundance is increasing. And again, toxicity varies, but I'll just point out again in June, higher cell count, low toxin quota per cell. And Beacon 42, we don't have any genotype data for Beacon 42 yet. This is just cell abundance at the top and then toxicity at the bottom. But in general, again, toxicity is decreasing with increased cell abundance. So if we look at the genotype dynamics among sites, right, we're just trying to gain an assessment of what's going on with Pyrodinium out there in the Indian River Lagoon. There's definitely temporal differences, right? Over time, which you have to consider, okay, based on time, you're going to have different environmental factors, right? The salinity is going to fluctuate, the temperature, and I'm not even going to talk about all those different parameters. But SXTA4 dominates, especially during the early bloom season. Early bloom, I'm considering March. Toxin trends, these are a little more difficult to see here in 4 different graphs, but overall you can see, I wanted to include this because you can see that Beacon 42, now granted, we only have May, June, and September data, but overall it has lower toxin content per cell than Diamond Bay and Pullover Canal in general. So looking at the relationship between cell abundance and toxin quota, you can see that in general, decreasing toxin content per cell, and it's going to increase as the cell increase in number out there. All right, so what is the function of these toxins? We've been talking about it. Saxitoxin is a potent neurotoxin, but you know what? These cells do not make the toxin to go after you. Saxitoxin and dinoflagellates evolved way before the human sodium channel, which is the target, which is how it affects us. And so for saxitoxin, one of the leading hypotheses is that it functions as a grazing deterrent or a defense from predators, right? The copepods and other zooplankton are out there in the water column trying to eat these dinoflagellates. And so they have this toxin, the copepod tries to eat them, it can have different effects on them, right? Different species it has different effects on. I've also done work that shows that the toxin may be functioning to help the cells with copper homeostasis. Copper is a trace metal. We need it, phytoplankton need it. If there's time, I'll get into it. But then there's other theories as well as to what the toxin is doing. Why are the algae producing it? What's its role in their lifestyle? Some suggested functions as a pheromone. Others found that when you look inside the cell, it's going to help with the structural organization of the DNA. If you look back at the structure of the saxitoxin molecule in general, you'll see it has a lot of nitrogen atoms. You are all familiar with nitrogen inputs in the Indian River Lagoon. At least you've heard a lot of nitrogen going in or different nitrogen forms. Well, could it be a form of nitrogen storage, helping them with these higher levels of nitrogen? All right, so these are some theories as to why it's producing it. I have to end soon, but I just want to try and link toxicity and bioluminescence in Pyrodinium bahamense. So now I've been talking to you about SXTA4 and toxicity. I want to speak briefly on bioluminescence in dinoflagellates in general, because both toxicity and bioluminescence are energetically expensive. The cell has to use up a lot of energy to produce the toxin or to produce this flash of light. And so with bioluminescence, as I said, there are non-toxic dinoflagellate species that are bioluminescent. Pyrocystis is one example. It's a big cell. It's like a millimeter. And I've done work with Dr. Edie Witter, and others have done work as well, showing that this bright flash from Pyrocystis, which is non-toxic, is bioluminescence may be functioning as a burglar alarm, in which you have the dinoflagellate and you have its copepod predator that wants to eat the cell, but it's going to flash. It's stimulated to flash by the copepod swimming currents, and it's going to attract a secondary predator, a fish that's going to come along, eat that copepod predator, save the dinoflagellate cell. Okay, that's a bright emitting cell, and we've done studies with Witter and Case did studies with cephalopods. We've looked at it with Pyrocystis and flame cardinalfish. All right, so that's one way of how the flash may be functioning. But then you also have dim emitters. So Pyrodinium and Alexandrium, both toxic, bioluminescent, they are more of the dim emitters. Their bioluminescence isn't as bright. And so with these dim emitters, this bioluminescence may be functioning as an aposomatic signal. It's warning the predator, don't eat me, I'm toxic, right? And so the predators are maybe learning to associate the flash with toxicity. Okay, so is that clear to everyone? You have these different species, they have different levels of bioluminescence. And so how is that flash functioning to protect them? All right, linking it in Pyrodinium bahamense. So trying to tie in the SXTA4 data and bioluminescence. Do you have cheats in the subpopulation? So both bioluminescence and toxicity are thought to play roles to help the cell function as a grazing deterrent. And recent work in Edie Witter's lab showed that there might be Batesian mimicry occurring within two different dinoflagellate species. Batesian mimicry, even if you're not familiar with the term, if you look at these butterflies, you might say, yeah, I know what that is. I don't know. Those of you that are familiar with the viceroy and the monarch butterfly, this is Batesian mimicry. The monarch butterfly has this bright coloration and it's toxic. The viceroy is the mimic here. It's colored just like the monarch, but it's taking advantage of the monarch's toxicity, which is then protecting it from the prey. So it's also warning the predators, oh, don't eat me, I'm toxic, when really it's not. So with Pyrodinium bahamense, do we have cells within the population, the SXTA4 minus, that are there, that are then helping to expand the bloom? Because as I said, toxin production is energetically expensive. Bioluminescence is energetically expensive. So if you can get rid of one, but still have protection from the group as a whole, is this loss of toxicity going to allow you to grow faster, to bloom, to help expand the bloom? And so what may be going on here, what we want to look at is whether STXA4 positive genotype or induction, repression of the SXTA4 gene is going to dominate at the beginning of the bloom, right? Where all cells can be toxic and bioluminescent. And that toxicity is helping them, whether it helps alleviate copper stress, however it's functioning, whether it's serving as a grazing deterrent. And it's going to help the bloom start to grow. And then as you get a lot more cells, right, and all of these flashes, and you have your predators in there, well, then are some of the cells going to be repressing the toxin gene, or there are some strains in that mixed population that are then able to grow that don't have the toxin gene, but overall, it's allowing the cells to bloom, right? March and April, you don't have as much bioluminescence, you don't have as much cells in the water. Then we get to June, where you get a lot of cell growth, you get a lot of bioluminescence, but you saw that toxicity was decreasing. So I know that my time is up. I was going to talk more about transcription, because that is very important coming into play. So if anybody in the Q&A wants to talk about transcription, I would love to discuss it with you all. I also just want to mention that we are working on a field tool to help quantitatively measure bioluminescence, because in Puerto Rico and in different sites, they measure it on a Viz scale. It's the bioluminescence is a 5, the bioluminescence is a 10. Well, that's very subjective, right? And so we need a rapid field tool to be able to go out and quantitatively measure the bioluminescence. And really, bioluminescence of Pyrodinium serves as its own visual tag. Hey, there's a Pyrodinium bloom occurring because I can go out to my dock and wave my hand through the water and see bioluminescence. And so eventually I would love to engage the public's help in this as well, being able to look at when the blooms are occurring, the abundance, the bioluminescence, the toxicity levels. And so I was going to talk more about intensity, flash intensity, sequence differences, but I'm going to stop there. So let me get to and I was going to talk more about toxin and copper homeostasis, but I'm going to end there, leave you on that note of bioluminescence and toxicity in Pyrodinium, and thank people that have been involved in this study with me, and hope that I've been able to tell you something of a story that's going on out here in our backyards, and to not be afraid of saxitoxin, since now you know how it works. And you can appreciate the brilliant bioluminescence of pyrodinium. All right, so with that,
Alrighty, so as Bree is walking around collecting the cards from the audience, would you like to tell us quickly about a time a manatee saved you in the Hall over
well, okay, if there's any FWC officials listening to this, I apologize, but I like to open water swim. Okay, I've done open water swim around Key West, and I was up here and I didn't get down to Key West for the swim this year, and I wanted to do this long distance swim in the open water. I love swimming in the open water. And the week that I was here, because I go back and forth between Virginia and Florida, the ocean was really rough. I was like, I'm going to do my long distance swim. All right, I'm going to sample the Hall Canal. So I had discussed this with Sunny. Where can I swim where I won't have to worry about the alligators, right? I'm in Florida, and she's like, where are you going to find a spot? So anyway, we decided on the Hall Canal, and I was just going to swim along the shoreline. But then, it's so shallow. So yes, out of the way of the boats and everything, totally aware of the boats. But I'm swimming down Hall Canal. Are you all familiar with Hall Canal? Well, it's beautiful up there, right? And you can't really see though when you're in the water. It's just green water, sunlight coming through. And so when I'm swimming back up, all of a sudden my arm hit something. And I was like, oh, I hit a rock. But then this gray shape emerged under me. And it was a manatee. And she stayed with me. I don't mean like she stayed there. I mean, my belly was on her. I was surfing with the manatee. And I was just like, who cares about swimming and my time at this point? This is so cool. I'm with a manatee. And maybe she'd leave, but then come back. And this went on for like 15 minutes. And I see these fishermen on shore, and I'm like, and they're looking at me. And finally, this one guy yells, and I'm going to tone down his language, but he's like, there's a huge behind you. And I said, what? And I looked and I bolted for the shore. And then I'm standing there, bare feet, on the scree. And the manatee's there, and I see the gator, and he's like, dude, I'm not even throwing my line in the water. And that thing started coming towards shore. So I scramble up on the bank and I start running down the road in bare feet. But that manatee saved my life, because then, I learned about the woman that in the neighboring town that was killed when she fell into a retention pond and 2 gators jumped her. And then I learned about the triathlete who was open water swimming, and his friend had a drone and caught his gator attack live there, and they got him to the emergency room. So I am so thankful for that manatee.
Wow.
And the next time I was up at Beacon 42 and I'm on the boat ramp filtering, here comes a manatee, snout up, wanting that water. That was my manatee. Anyway.
Example has one of the highest alligator densities of anywhere in the U.S., North America. It's not a place to go.
I know, I'm so disappointed.
No way.
It's fairly safe.
No canal, but
All right, so we'll move on to some questions now. Thank you for that story. Why are pufferfish susceptible to the saxitoxin?
yeah, can you see?
Yeah, why are pufferfish susceptible to bioaccumulating the saxitoxin?
Yeah, I'd have to think back on my pufferfish knowledge, which is not a lot, because pufferfish,
Small mussels.
okay, but as opposed to, other fish would eat the mussels as well, I think. So I guess what I'm thinking is, do they have a better affinity? But I don't know. So anyway, they're eating what's been accumulating the toxin. And so saxitoxin then stays in their tissues.
So we had some locals who grew up on the Indian River Lagoon noticed that until after 2000, pyrodinium was strictly seasonal. They're saying red bloom in August through mid-October only, but now they're seeing the red blooms from June through November, specifically between the Bennett Causeway and the Native Causeway. Is there a reason for this extension?
So I would say, look at the water temperatures, because overall, yep, climate change, it's happening, right? Harmful algal blooms in general are increasing worldwide. The Indian River Lagoon, I don't know how, pyrodinium is more of a tropical species. It likes warmer waters. It likes higher salinities. So if those things are changing, plus we do have more nutrient inputs going into the Indian River Lagoon, right? You all know that, and you're getting these blooms that then kill off the seagrass. Well, what are these nutrient runoffs? Nitrogen, phosphorus, What are pyrodinium? They're like little plants. So it's like they're getting this fertilizer to grow more. So
Is the pyrodinium opportunistically toxic, such as microcystic?
So what do you mean by opportunistically toxic? Because- Okay, they could specify, well, so when they do culture studies, yes, toxicity varies with the growth stage. And that's why they do a lot of lab studies as well with these toxic species and strains to understand how the toxin content and composition is going to change based on the different nitrogen sources, the different levels of phosphorus, salinity, temperature. So is the toxin increasing or decreasing based on nutrient availability, temperature, salinity? Probably a combination of all. But when you say opportunistic, it's also important to recognize that there have been some great studies done with Alexandrium that shows copepod cues. Copepods emit these chemicals that induce toxin production in Alexandrium. So they produce more toxin because they sense the copepod cues. So there are so many factors that, or so many different reasons as to what could be going on.
Has there been any consideration of controlling the pyrodinium by releasing a virus?
Oh, I don't know. That's an excellent question. I don't work with the government and the governing agencies in terms of what mitigation strategies they're considering.
Does the toxin flush out of our systems, or does it build up over time?
Oh, it would break down over time. Yeah, once it's bound to your sodium channel. But there are different treatments for it. So it will be broken down and washed out over time. And that's what it does in other organisms as well. Their enzymes can like change the shape of it.
And then this is the last question that we're going to have time for today. How much do water temperature or depth have an effect on the toxicity?
So I don't have the answer for that. Like I said, water temperature, they do like warmer waters. But if you look at, like I use Orca's Kilroy data, and if you look, even at 18 degrees, there's still cells in the water. They like 20 to 30 degrees Celsius better. So water temperature, huge factor. And in terms of depth, dinoflagellates in general are going to vertically migrate throughout the water column. They're going to go lower at night to try and get some nutrients, come up higher during the day for photosynthesis. So I don't know the extent that vertical migration affects Pyrodinium bahamense toxicity. All right. Thank you all.